I was looking upstairs, and I saw William made a Quizlet, and we don't see them unless we're in Vegas. So I'm like, oh, is that one? Who's mine is? Oh, did you hear? I was so upset. That whole, I think, stole my chance. Well, I mean, I don't understand. It's pretty easy. I'm not on your own.
Okay, testing. 1, 2, 3. Tia, I'm gonna assume you can hear me. If not, please feel free to message me or something like that.
I can hear you.
There we are. That works. All right. Okay, that works. And that works. Okay. Good, 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 good. Okay, back to Here comes the coffee crew.
Do you like it? Or you can- Yeah.
Good morning, everyone. So housekeeping first, to reiterate, the Wednesday section will have lab this Wednesday. That's to catch you up to the Thursday section, who had lab the week of Arbor Games. The Thursday section, of course, will not meet, but the Wednesday section will meet. Now, Thursday section, folks, you guys at tubes, you're probably going to have to read. So it would probably behoove you to come in and read your tubes and plates and so on and so forth, so that you have that data. So that means if we put the tubes and plates out, we can then put reagents out so you can have time to come in and just read the plates, put some phenyl red in your tube, see if they're acidic or not. Read your starch plates, read your spirit blue plates, and so on and so forth. Wednesday group will do that in lab. The Thursday group, take some time on your own and find time to do that. So we will have lab Thursday, but we will not have lab on, sorry. We will have lab on Wednesday, we will not have lab on Thursday. Secondly, I hope you all got the good news by Dr. Baldwin. The neurologist said there is no objective evidence he had a stroke. None of the scans they did, and they scanned him 3 times, could detect any blockage or any remnants of blockage. Furthermore, he has no neurological deficits whatsoever. It looks like he had a bad migraine headache. Turns out those run in his family. So he's had bad headaches before. He always thought they were sinus headaches. Turns out he probably gets migraine headaches. So Bruce will come home today. So I saw him Friday, Saturday, and then I wasn't able to get to the hospital Sunday, so I called him. He's doing great, really doing great. His wife's doing great too. So, which is progress, because boy, Friday was not good. So, we're very thankful for Bruce's recovery. So, let's all give thanks. We are talking about gene expression today. Gene expression models are not simple, they're complicated. I'll show you some videos. You're going to have to really just rehearse it in your head 2 or 3 times, 5, 6, 7, 8 times before it really starts to sink in. It's not simple. There's a lot of moving parts. And when you learn something with a lot of moving parts, it's hard to keep them all straight. It's kind of like cardiac pathologies. Oh, is this one systolic? That a systolic rumble, or is that a diastolic rumble? Is a crescendo, decrescendo, or which one supposes paradoxes? And was he EKG look like? You know, it's just very hard to keep them all straight, isn't it? So it takes several passes at the material. It's going to be that way today. Let's start with a word of prayer. Lord, thank you so much that Bruce is doing better. Thank you that Michiko is doing better. Thank you, Lord, for the power of a loving body of believers who surrounded them, both of them, and supported them. We pray Bruce is able to go home early today, that he gets all released from the hospital, that he's able to sleep in his own bed and actually sleep a full night and not have people futzing with him through the night. And Lord, we pray for full recovery and that he can pass through our doors again soon. Thank you for the students here and all their hard work. Thank you for our bowling team. We had a really good tournament. And now we pray that we now focus on the work ahead of us today, gene expression. Wake us up and help us learn. Pray to your glory in Jesus' name. Yeah, bowlers, heard you guys didn't do too shabbily. Yeah? Well, don't show so much excitement. I heard the second place, I heard, is that? No? For JV, yeah. Guys, that's. Hello. No, that's second place out of how many teams?
I don't know, like, no way.
There's several teams. That's not bad. Yeah.
Oh, Davey, there was-
Okay, you wanted to win. Fine, you wanted to win, but
Davey didn't get. Varsity.
Okay, so varsity stumped it up, fine, but JV getting second, that's, well, you had one good day though. Come on, you gotta count your blessings there, hon. Be so hard on yourself. No, well done, bowlers. Well done, all of you. I'm very glad to hear that things went well. I heard a rumor things were going decently, and then Gracie said, yeah, maybe took second, so. Yeah. DNA, RNA, three big differences. So DNA uses 2-deoxyribose as its 5-carbon sugar. That means the number 2 carbon, instead of having a hydroxyl group, has 2 hydrogens. That's the only difference. RNA has ribose as its 5-carbon sugar. Bases. DNA has adenine, thymine, cytosine, guanine as its bases. RNA has the same bases except that thymine is replaced by uracil. Uracil. Chemically, uracil and thymine are very similar. A methyl group separates them. In fact, thymine is made from uracil in the cell. But uracil is the base that you find in RNA. If there is thymine in RNA, it's put there after it's made. It's put there post-transcriptionally. Thirdly, DNA is a double helix in cells. RNA, unless you're a double-stranded RNA virus, or unless you're a plant where they do these oddball things with double-stranded RNAs virtually are virtually non-existent in cells. If they do exist, they're usually intermediates and synthesis intermediates of certain viruses. And double-stranded RNAs inside human cells induce interferon. What's interferon? Anybody know what interferon is? Of course not. We haven't studied immunology yet. When we get there, you'll learn all about interferon because it's very important in the immune system. Interferon is an antiviral system. It induces the antiviral system in your cells. And double-stranded RNAs will induce interferon, and they'll induce it quite powerfully. So RNA is usually single-stranded. Bits of it might fold back upon itself and form secondary structures. Some of those secondary structures are pretty important for its function. So it's not a double helix like DNA is. So those are 3 main differences. So if I ask you on a quiz about the 3 main differences between DNA and RNA, think you can list them? Your confidence truly inspires me. Oh, you believe so? Thank you. It'll take a couple passes at this maybe to get to really imprint it in your memory, but those are the main differences and they are important. So when it comes to RNAs, there are regulatory RNAs that are listed here, small interfering RNAs or siRNAs, riboswitches. Riboswitches are used for regulation. A little bit in eukaryotes, more often in prokaryotes. Antisense RNAs, which are found in both prokaryotes and eukaryotes, and microRNAs, which are used mainly in eukaryotes, but also get used in prokaryotes. We talked about primers that are used for DNA synthesis. Those are used in both eukaryotes and prokaryotes. And then ribozymes, RNA molecules with enzymatic activity. So the other 3 main RNAs used in cells are, first of all, ribosomal RNA. Ribosomal RNA plays a structural role in ribosomes. And the ribosomal RNAs of bacteria are of a certain type. So if we use is my light on? Okay, light, you can come on now. There you are. Okay. So when it comes to ribosomal RNA, which is designated as rRNA. Remember, in bacteria, their ribosomes are smaller than they are in eukaryotes. So in prokaryotes, you have the small ribosomal subunit. The small ribosomal subunit in prokaryotes has a 16S ribosomal RNA. The small ribosomal subunit in eukaryotes has an 18S ribosomal RNA. It's bigger than the one you see in bacteria.
Is that a 5 or an S? Right by the-
That's an S. The large ribosomal subunit now remember, the small ribosomal subunit in prokaryotes is a 30S small ribosomal subunit. It's bigger in eukaryotes. In eukaryotes, it's 40S. Then when we talk about the large ribosomal subunit, this is a 50S large ribosomal subunit. In prokaryotes, it's a 60S large ribosomal subunit in eukaryotes. In prokaryotes, you have a 23S ribosomal RNA in the large ribosomal subunit and a 5S ribosomal RNA. In eukaryotes, you have a 28S ribosomal RNA, a 5.8S ribosomal RNA, that's a 5, and then also a 5S. So these two seem to be similar. These two are definitely similar. This is the oddball. Prokaryotes don't have any RNA like this one in their ribosomal subunits. These provide structure for the ribosome, but this large ribosomal RNA is a ribozyme. This has enzymatic activity, and this is the entity that forms peptide bonds on the surface of the ribosome. Come on, get in focus, you. Okay, so it's a ribozyme. Ribozymes are RNA molecules that have enzymatic activity. And these large ribosomal RNAs, both in prokaryotes and in eukaryotes, they catalyze the formation of peptide bonds between the amino acids that are on the ribosome surface. So those are your ribosomal RNAs, rRNAs. Page?
Is that small?
Those are the subunits, the size of the subunits of each ribosome. The ribosome has two subunits, a large and a small. The 30S is the size of the small ribosomal subunit in bacteria. The 40S is the size of the small ribosomal subunit in eukaryotes. 60S is the size of the large ribosomal subunit in eukaryotes. In prokaryotes, the size of the large ribosomal subunit is 50S. And then the size of the ribosome in bacteria is 70S. The size of the ribosome in eukaryotes is 80S. 80S. Transfer RNAs ferry, F-E-R-R-Y, ferry amino acids from the cytosol to the ribosome for the purpose of protein synthesis. All transfer RNAs have CCA at the 3' end. That CCA is added after the tRNA is synthesized. The 3' end is the end that to which the RNA is attached. Then the tRNA forms several different secondary structures, and it forms this clover leaf-like structure that is a result of self-hybridization within the molecule. This self-hybridization forms these different hairpin loops. Now, the hairpin loop opposite the acceptor site, acceptor for the amino acid, is called the anticodon. The anticodon loop, and you have 3 bases here. That's the anticodon, those 3 bases. Those 3 bases are the bases recognized by the messenger RNA that's held by the ribosome. And you can see the messenger RNA here. These 3 bases here, the codon, will anneal to the bases in the anticodon loop. The secondary structure is essential for the activity of the tRNA. Absolutely essential. Without the secondary structure, it's not going to work. In fact, many, many years ago, a researcher by the name of Karana in his lab, they showed, say, if you put a mutation here and you prevent this loop from forming, the tRNA doesn't work very well. However, if you put a compensatory mutation over here, so now these 2 bases can still hybridize with each other. Say, for example, you put a cytosine right here, that tRNA doesn't work as well. Now, if you change this base over here, make it a guanine, now that tRNA works again. So the formation of the hairpin is extremely important for the function of the tRNA. So the secondary structure, the cloverleaf structure, it has to have that in order to properly work. Not only does it have secondary structure, this particular RNA has tertiary structure. That is, it has a structure within 3D space. And here's the tertiary structure of the tRNA. These 2 loops kind of clap their hands like that. The anticodon loop then goes like this, and the amino acid attachment site goes out like this. So the molecule is shaped kind of like an L. So, that L helps it fit on the ribosome. Messenger RNA is up here, but the catalyzation of the peptide bond will be done down here. This tertiary structure is dependent on the secondary structure. And this tertiary structure is essential for its ability to bind the ribosome and assist in translation. Messenger RNAs are made from stretches of DNA and they're used to encode a protein. They're used to encode a protein. So this emphasizes the differences between ribosomes in bacteria and eukaryotes. Let me emphasize that the ribosomes found in mitochondria and chloroplasts are bacteria. They have a bacterial way of doing things. They're bacterial size. They're bacteria. Said all that already. All right, so with this in mind, let's watch a video that gives you an idea. Oh, no, I don't want that. I want this one. Yeah, that one. Okay. We are going to watch a video about protein synthesis. Turn up the. Okay, hopefully this works.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
There, get rid of that. Oh, you know what? Let's take a look at something here. All right, so the messenger RNA has a front 5' end and a 3' tail. Remember, we said the nucleic acids have polarity. They have a front end and a back end. The front end is the 5' end. The tail is the 3' end chemically. Your book has some good pictures of the 5', 3' end in the molecule. Go ahead and review that if you don't completely remember what that is. The ribosome is going to start usually towards the front of the molecule. The very first codon that the ribosome will recognize is always an AUG. AUG codes for the amino acid methionine. Methionine. And there's a special methionine tRNA that's used as the start of translation. Now up here, there's a sequence. This sequence is called the ribosome. Binding site. The old term for it is Shine-Dalgarno sequence, named after David Shine and Frank Dalgarno, who discovered it. But the ribosome, the 30S ribosome, is able to bind to this site because the 16S ribosomal RNA has a sequence that is complementary to this sequence here. So that ability of this part of the messenger RNA to hybridize, to anneal to this tail end of the 16S ribosomal RNA, that essentially is going to glue the messenger RNA to the ribosome. And then the very next AUG, right after the ribosome binding site, that's going to be your start site for translation. And then the ribosome will read this RNA 3 bases at a time right after that. There are 3 particular basic 3 codons that don't code for any amino acids. Those are UAA, UGA, UAG. These don't code for any amino acids, and these are all stop codons. When the ribosome comes to a stop codon, instead of the next tRNA binding, proteins called release factors. Release factors bind, and they essentially take the ribosomal subunits and dissociate them. So the upshot of all this is, first of all, the messenger RNA is read 3 bases at a time. Those 3 base units are called codons. They recognize specific tRNAs. Those tRNAs have in their anticodon loop a 3 base sequence called an anticodon that is complementary to the sequence in the codon. And therefore, the sequence of the protein, the amino acid sequence of the protein, is reflected in the sequence of the messenger RNA. And since the messenger RNA is made from the DNA, the sequence of the protein is directly reflected in the sequence of the DNA that codes for that messenger RNA. All right, that was a mouthful. Let me pause right there. Make sure everybody's on the right page. Milena?
Can you explain the stop codon? Or
Sure.
just
Once translation starts, each next triplet of bases will code for a particular amino acid. And you'll code for methionine, threonine, glycine, isoleucine, phenylalanine, proline, cysteine, your mother-in-law, blah, 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 whatever. Okay. Until you get to one of these three codons. When you get to one of these, the tRNAs will all look at each other and go, do you go for that? I don't go for that. How about you? You go for that? I don't go for that. So since none of these code for a tRNA, release factors will bind. And translation, they will dissociate the two subunits of the ribosome and the protein and the messenger RNA and so on and so forth. So these are stop codons. When the ribosome gets there, translation stops. That's why they're called stop codons. Catherine? Those represent the front and the back of the nucleic acid. So nucleic acids so there's a phosphate here, and then this particular one binds to another phosphate, binds to yet another. This part here, so this is the 5' carbon. That's the front. That's the front of the nucleic acid. This down here, when you get to the last base, that's the 3' part of the molecule. That's the end, the tail. So that's what essentially these represent. This is the front of this molecule. This is the tail of this molecule. This is the front of this molecule. That's the tail of this molecule. So 5' and 3' represent the front and the back of nucleic acids. Nucleic acids have polarity. They're not the same at both sides. The two sides are different. And messenger RNAs are read from front to back. They're also made from front to back. That polarity is important. So as we said, you have players in translation, and those players, they include, first of all, tRNAs. Those tRNAs are charged with an amino acid. Because they're charged with amino acid, they're able then to go to the ribosome, and if their anticodon is right there, their anticodon will hopefully recognize the sequence of the, that's the thing. Their anticodon sequence will recognize the codon sequence here in the messenger RNA. So here's the other player in translation. That's the messenger RNA. And it gets made by protein coding genes. It's transcribed, and then it is engaged by ribosomes and translated. And within the ribosome, you have ribosomal RNAs along with ribosomal proteins. Oh, that's terrible, but you know what it means. It's hard to write with a mouse on there. It's not very good at it. Now, each codon specifies for a particular amino acid. Yes, Morgan, I see that that's difficult. There. Better? You're welcome. And this code by which certain codons essentially will determine a certain amino acid is put into a protein. This particular code, genetic code it is called, turns out to be universal. Did you guys ever wonder why human insulin can be made in bacteria? Because the human gene works inside a bacterium. Because the genetic code that the bacterium uses to translate that protein is the same code that our cells use. So the code is relatively universal with only a few exceptions. And here is an example of that code. So AUG, as we said before, that's a start codon. Some of these, as you can see, they're different codons, more than one will code for certain amino acids. Arginine, in fact, as you see here, arginine is coded for by these two and by these four. So arginine is coded for by 6 different amino acids. Others are coded just by one. Tryptophan, for example, and methionine are just coded for by one codon. And then others, leucine. Leucine is a bit of a pig here. It's 6 different codons code for it, and what have you. So this is the genetic code. This is how the ribosome is going to read the codons in that messenger RNA. This will determine the primary structure of the protein that's made. Now, also there's this term redundancy. Redundancy means that more than one codon codes for the same amino acid. Some degree of redundancy means that you can actually have changes, mutations in some of these codons, and they won't change the type of amino acid for which you code. So that essentially makes it so that certain mutations actually won't damage the type of protein you make. Now, there's a problem with redundancy, however. Redundancy means you're going to need lots of different tRNAs to be able to read each of these different codons. Well, the cell has a solution for that too, and it's called wobble. Wobble, just like weevils wobble, but they don't fall down. Wobble means that the first 2 nucleotides that read the codon, now remember, this is something that's through the geometry of translation. When you consider the messenger RNA, say, for example, you have a tRNA. So let's say this is, so this is 5 prime, 3 prime. The tRNA then is going to be reading this 5 prime, 3'. Its anticodon is going to go like this. Like that. So this will be C and G. But this particular base here, that's going to be further away from that one. This is kind of linear sitting on the ribosome. This isn't. So the type of base pairing you can get between these two bases, essentially, I've got this backwards, I'm sorry. It should be between these two. The type of base pairing you're going to get, let me redraw that. Say, for example, so this will be, say, 5', 3' in the tRNA. This is a messenger RNA. This is the tRNA down here. So this anticodon this is not linear, it's a loop. Like that. This coding here in codon number 3, the number 3 position, because there's further distance between this base and this base, you don't have to have a G there. You can have a different type of base there and you'll still have good pairing. That is called wobble. That means that the type of interactions that you have between these two bases is less restrictive. It's more permissive because of the geometry of the interaction of the tRNA with the messenger RNA. Therefore, wobble means that you have variation in the number 3 position of codons. And if you look at the genetic code, you can see a lot of these vary at position number 3. But the same tRNA reads all 4 codons. That means the number of tRNA genes that you have to encode is not very high. So you can get a lot done with a smaller number of tRNAs. And that's what wobble does for you. So you get the benefits mutationally, where the mutations in the number 3 position won't tend to cause huge changes in the sequence of the protein, amino acid sequence of the protein. And you don't have to encode as many tRNA genes in order to read the genetic code. So that means you can look at the sequence of DNA of a gene, and you'll be able to directly predict what the protein sequence will be, because essentially of this mechanism of translation. And that's essentially how people are able to get protein sequence, even though they haven't sequenced a protein. Sequencing a DNA molecule is much easier, much cheaper to do than sequencing a protein. Yet, you can sequence DNA and get protein sequence information out of it for this very reason. All right, let me stop right there. Make sure we're all on the same page. All right. Now, when you make a messenger RNA, you don't have to have one lone ribosome on that messenger RNA. Another ribosome is not going to get on that RNA until this one finishes. No. That's not the case. Once a ribosome gets out of the way, second ribosome can engage it very easily. Therefore, it's very common for messenger RNAs to have multiple ribosomes engaging them at once. This sort of structure is called a polysome. A polysome. And you see it right here, polyribosomal complex. This is illustrated really nicely in this thing. This is called a Miller spread. This comes from Art Miller's lab at University of Chicago. So this is a stretch of DNA. This very famous picture was actually taken by Barbara Hamcallo in his lab. Barbara Hamcallo was one of my professors at UC Irvine. And you can see RNA polymerase molecules making the RNA. And what is attached to all those RNAs? Ribosomes. The longer the messenger RNA, the more ribosomes are attached to it. So it turns out that once a messenger RNA is engaged by a ribosome, many ribosomes can engage it. They all have to start at the same place, but you can have polysomes, and in fact, polysomes are the rule. Now, there's a big difference between prokaryotes and eukaryotes. In eukaryotes, you make a messenger RNA, it gets processed, it gets exported to the cytosol, then it can be translated. Do you have a nucleus in prokaryotes? No. So do you need to export RNAs to the nucleus, to the cytosol? No. So in fact, RNAs can be translated even while they're being transcribed. This is called coupling of translation and transcription. Coupling of transcription and translation. Very common in prokaryotes. And as we will see a bit later, it's going to play a role in gene regulation in prokaryotes. We went through the steps of translation already. I'm not going to go through this again because you showed the video. Okay, now in eukaryotes, if you sequence genes in prokaryotes, you have a one-to-one relationship between the sequence in the DNA and the protein sequence. That's called collinearity. Collinearity is the rule in prokaryotes. In eukaryotes, a funny thing happens on the way to the bakery. When you sequence protein coding sequences in eukaryotes, they've got these sequences that interrupt them. These sequences are called introns. Introns. Turns out, when you make messenger RNA, when you make the messenger RNA, those introns are spliced out by a process called RNA splicing. Only when the RNA is properly spliced. So you pull these bits, introns out, and you ligate these exons together. Can the RNA actually be exported to the nucleus for translation? So messenger RNA encoding genes, protein coding genes in eukaryotes are often interrupted. They're so-called split genes. And as you go from unicellular to multicellular organisms, the genes start having more introns. So yeast genes, for example, don't have very many introns. Fruit fly genes have more introns than yeast genes. Human genes and mouse genes hit the mother lobe. Most of the sequence is intron. The stark majority of the sequence is intron. So they have lots of introns and they have big introns. And that seems to be the theme as you get to multicellular organisms. They have more introns and bigger introns. Bacteria do not have introns in their protein coding genes. Which of the following types of RNA contains hairpin loops and carries amino acids to the ribosome for translation? Lexi?
What's the purpose of insurance? Or is there
I
not?
will give you an educated guess.
Okay.
You ready for an educated guess? How many different types of muscles do fruit flies have? Hundreds. They have muscles for direct flight muscles. There are actually no muscles in the wings. The direct flight muscles are the thorax. The direct flight muscles do this to the thorax. That's what causes the wings to flap so hard. Those are called indirect flight muscles. Then they have direct flight muscles that actually help turn the wing. It helps the fly do acrobatics in the air. Speed up, slow down, turn, what have you. Then they have muscles for unrolling the proboscis. They have muscles for breathing. The males have muscles in what's called the sperm pump. You can guess what the sperm pump does. They have jump muscles in the legs that help them launch themselves into the air. They have muscles in the antennae that direct them in different places. All those myosin molecules are different for every one of those. Guess how many myosin genes the software has? One. How's it able to make so many myosins from just one myosin gene? Differential splicing. It has bucket loads of introns. Some of them are ignored, some of them are not. Some of the myosins are shorter, some are longer, some have this piece, some have that piece. They mix and match this and that through regulating what kind of splicing they do in whatever types of muscles they have. So these cells probably have introns so that you can make lots of different protein products from the same stretch of DNA. Remember pathophys you studied, did you remember studying about migraines? You remember studying about the calcitonin gene related protein? That's the, when we talk about the new drugs in farm that are used for migraines, that's the peptide that they inhibit. They inhibit the CGRP. CGRP protein is made from the same gene as calcitonin. It's differentially spliced. So, same stretch of DNA, you make lots of different proteins. That's my best guess. Okay, let's take a break. Take 5, come back, get back in the saddle.
But it was.
They got rotation, but they're not doing so much of the hours.
So, yeah, I mean, they were.
Yeah.
They don't.
So the game is changing.
One girl had a red. They're out in back. Yeah. And rather than being infected and just. No, she didn't have it, like, her thing off, and she made that. That was.
So it was a cheap shot.
Yeah. Twice, so she got a yellow, yeah, earlier.
Twice?
And then she got a yellow.
I thought it was flagrant to see that.
No, I mean, if you get 2 yellows, you're good with red. Yeah, but the first one wasn't bad, the second one was kind of like showed the red card, the first one was kind of like, she didn't want to take her out and have the L and.
That was, sorry.
Yeah. And even their stands, like their stands were really rude. My mom was watching the live stream and they ended up shutting off the audio because they didn't hear anything. My mom's like, why is that audio going on? Yeah, it's probably because the fans were so because they were getting like mad when they scored like later in the games and scored again. So yeah, they were Yeah, I was.
Yeah,
We're getting.
I watched Taylor over here and I noticed that. I don't play soccer, so my opinion doesn't matter. But it seems to me there's a difference between playing tough and playing rough.
Yeah.
You can play tough, even a little rough, and then there's just a line. The same thing where you're doing stuff, I mean, they were doing stuff. If you've got the ball, someone that you got, they were doing stuff like this. They were just putting people on the wheels of the stuff like that. And that seems maybe, that to me doesn't seem to be part of the game. Aggressively, blocking somebody out, that's part of the game. That's, being tough, but there are certain things where you're hip checking people, that's probably part of the game to some extent too, but when you're doing it like part of that, and you're swinging around elbows and stuff like that, that's, trying to block that when you're swinging your elbows and aiming something to draw, you're trying to fix it with that.
They were doing a lot of stuff after, like, not even having the ball from the girls, just like check. Like a lot of them are in the beginning of the speed, we're getting like check right after the play, the ref is here and
You're trying to take it.
yeah.
Yeah.
We're not very good at this. Yeah.
Yeah, we've seen guys, they do it.
Right. Last year, it's, yeah, they're not better, they're just, they're just, yeah.
Technically, that's not funny.
Not, yeah, but they give us a hard battle. We give them a shot.
So that's not really good.
They're very. Yeah, like there's. When they get on the ball, like, but.
But that's not mentioned.
I don't know what to do. They can individually say, yeah, somebody mentioned. No, they're trying to. Yeah, like we beat them, and like our offense, like towards the second half, we like turned them down and like got them really like infuriated. That's how they were getting a bit like. Yeah. But.
What was that game last year? I watched almost the whole second half. No, the goalie. Schwimmer scored like 2 goals, and then it came out, and the ball was literally at the opponent's end almost the whole time. Schwimmer never scored.
Oh. That whole.
That's-
Okay. Was it last year you said?
Yeah, the goalie was good.
Tactical.
Yeah,
Even the one not big part of the bus.
yeah, and then the goalie.
Yeah, because they can take part every act, so they just spend, take part in the
Just like in football. Yeah. Okay, folks. I neglected to mention that our lady soccer won this weekend, so congratulations to lady soccer. They played Taylor, who's besides IWU has probably been the second biggest nemesis, I think, for Spring Arbor, and Taylor always plays us tough. They have a bit of a reputation for being physical. Yes, meanies, that's the term that's used sometimes.
Just the fans are mean.
Yeah, the fans are mean too. I have to admit, when they come here, our fans are not necessarily nice to them per se. Yeah.
Yeah.
Yeah, those guys need to give it a rest sometimes. Yeah, it's all right. I mean, nice to be loved, but yeah. So your book has this table that compares and contrasts bacterial and eukaryotic translation and transcription. Please keep in mind that messenger RNAs in bacteria will have more than one ribosomal binding site and more than one translational start site. So messenger RNAs for bacteria can code for several genes, whereas messenger RNAs from eukaryotes code for one protein, only one. Whereas bacterial messenger RNAs don't have to, but they can code for multiple proteins. This particular situation is called polycistronic. Polycistronic in bacteria. In eukaryotes, it's monocistronic. Monocistronic. You have coupling of transcription and translation. In bacteria, you do not. Transcription, translation, those two processes are physically separated by the nucleus. And eukaryotic genes are interrupted by introns. That's not the case for the most part in bacteria. So now we get to this whole thing of bacterial gene regulation. Now, I want to try to define for you this thing called an operon. This is not going to be simple. So I'm going to have to define it for you and then probably define it again because I'm pretty sure you won't get it the first time because it's complicated. That's not a commentary on your intelligence. It's just the complexity of the material. So here's the definition of an operon. So let's say this is a stretch of DNA in some bacterium. Now, this is the promoter. The promoter is the binding site for RNA polymerase. So RNA polymerase will bind here and it will make a messenger RNA that encodes this gene product, this gene product, and this gene product. One messenger RNA encodes these three proteins. All right, you with me so far? Let's say these are three genes that you don't need all the time. It would be foolish for the bacterium, therefore, to express these genes constantly. Make much more sense for it to only express these genes when they're needed, right? So when it comes to promoters, there's something you should know about promoters. Some promoters are strong promoters. By strong promoter, it means RNA polymerase binds there often. And because it binds often, it can initiate transcription often. Therefore, it makes lots of messenger RNAs. Therefore, you get lots of these proteins being translated within a period of time. Other promoters are weak, meaning RNA polymerase does not bind to it tightly, meaning it only binds and initiates transcription occasionally. You don't get many messenger RNAs made. You don't get many of these proteins translated. You have very few of these proteins. So what if you have this situation and you have a weak promoter, but all of a sudden you need these gene products? What do? You induce them. You induce transcription. And if you do induce transcription from this promoter, you increase the amount of all three gene products. How did you induce all three gene products? By virtue of the fact that they're clustered together. You cluster them together so that you make this one RNA, and that one RNA is subject to a common mode of regulation. That is an operon. A cluster of genes subjected to a common mode of regulation. A cluster of genes subjected to a common mode of regulation. So that if you turn on just this gene here, you turn on all three actually. That's an operon. I see the blank stares already. I told you this was complicated. Paige? This cluster, gene cluster, that's an operon. The genes are often functionally related, not always, but they usually are. And because they're clustered and because you make this polycistronic messenger RNA that encodes all three proteins, by upregulating, by inducing the transcription of this one RNA, you upregulate the production of all three gene products. That's an operon. Okay, Devin, I see the smoke rising. I'll answer Melena's question first since she raised her hand, then I'll come back to you. You look lost, so I'll come back to you and see if we can get you unlost. Melena, go ahead.
Either be strong or weak.
Right. Promoter's a binding site for RNA polymerase. Now, RNA polymerase doesn't bind all promoters equally. Some promoters have a sequence that RNA polymerase really likes. Other promoters have sequences that RNA polymerase can take or leave. It's kind of like in the DC when they have, roasted Brussels sprouts. Some people really like roasted Brussels sprouts. Others people, you can't take them or leave them. Some of you probably mainly leave them. I personally like roasted Brussels sprouts, but some people, pizza. Yeah, a lot of people like pizza, which is one of the reasons why they started a pizza bar in that DC, because students requested it. They like pizza. Some promoters are like pizza. They're strong. Others are like roasted Brussels sprouts. They're weak. So it depends upon the strength with which RNA polymerase binds the promoter. And it depends on the sequence. I don't want to get into the sequence of it. That's beyond the scope of this class. Maybe in genetics, Aaron Wyman will regale you with stories of strong and weak promoters, but I will not go into it here. Lexi? Strong promoters don't need to be induced, they're already strong. You already get lots of transcription from them. If anything, you might need to shut them down.
Okay.
And that phenomenon is called repression. We'll talk a bit about repression here in just a bit. Okay, Devin, you have a question now? Do you understand this perfectly? You don't know what to ask? Well, why don't we look at an example of an operon and see if that helps? Shall we? Yeah, I told you guys this is complicated. Nobody believed me. Now, do you believe me? So operons are found primarily in bacteria. There are a few eukaryotes that have something approaching an operon. The worm, Caenorhabditis elegans, is the one that has something approaching an operon. They're not like what you see in bacteria, though. You also get clustering of genes in bacteria, in eukaryotes. For example, in humans, there's the globin, alpha globin gene cluster, and the beta globin gene cluster, but they're not operons. Operons exist in the archaebacteria and in the eubacteria. They're clusters of genes that are usually functionally related, and their expression is coordinated. They're regulated as a unit. Now, operons can be induced or they can be repressed. Very often they're a combination of the two, induce and repress. Now, some operons are repressible. They're repressible because they're turned off by the product that's made by the enzyme. Therefore, if you make too much of it, shut this off. Amino acid biosynthetic operons are good examples of these. So the tryptophan operon, for example, the tryptophan repressor shuts down the expression of the tryptophan operon. That repressor only binds to its regulatory site if it's bound to tryptophan. So only if there's more tryptophan than the cell can use will the tryptophan operon be shut down. And that's an example of a repressible operon. And Lexi, this actually gets to your question. So the tryptophan operon has a pretty strong promoter. So you make those enzymes at a pretty constant routine level. When you have too much of it, then you shut it off. Now, that's an example of a repressible operon. Then you have catabolic or inducible operons. Catabolic or inducible operons. When the substrate for the enzyme is not present, the operon is not expressed. Or if it is expressed, it's expressed at extremely low levels, so-called basal levels. When that substrate is available, then the operon is turned on, and that's called induction. So these are inducible operons. And the first of these that was worked out in any detail was in the 1950s by Jacques Monod and Francois Jacob, two French chaps. Okay, there's an Englishman who's on the paper too, but it's called the Monod and Jacob model, essentially. And this is the lac operon. So it was observed that when you give cells sugar, you give them glucose, the enzymes for degrading lactose, for metabolizing lactose, they're nowhere to be found in the cell. You can't find them. And if you look at the genes that encode those lactose utilization genes, those lactose utilization gene products, those messenger RNAs, they're not made. So that set of genes, they're off. However, once you give the cells lactose without any glucose, now those genes are turned on, and they're turned on 100-fold. So that's an example of induction. Now, in the cell, the lactose utilization genes are clustered. You have 3 genes. They're called the LAC-Z gene, the LAC-Y gene, and the LAC-A gene. The LAC-Z gene, the LAC-Y gene, and the LAC-A gene. The LAC-Z gene encodes this protein we talked about in lab, beta-galactosidase. It's the enzyme that degrades lactose into galactose and glucose. That's a product of the LAC-Z gene. The LAC-Y gene is the lactose permease. Permease. Now, what's permease? What's a permease? Permease brings lactose into the cell from the outside. It brings lactose into the cell from the outside. The LAC-A gene seems to be involved, it's called the transacetylase, seems to be involved in detoxifying certain types of plant metabolites that are kind of lactose lookalikes. Nobody still to this day is completely sure what the transacetylase does, but that's the best guess. So these 3 genes are clustered, and without lactose, they're off. And the reason they're off is that there is a regulatory site called the operator. The operator is right next door to the promoter. And when there's no lactose, the operator is bound by a protein called the lactose repressor. Lactose repressor. And the lactose repressor prevents RNA polymerase from binding to the promoter and initiating transcription from that site. So you've got a weak promoter, and you have a repressor that's sitting on the operator and telling RNA polymerase, oh, no, not this. You go away. Go away. No, I say go. You, other side of the cell. Go, be gone. So you get very, very little messenger RNA to make. And these gene products, therefore, are pretty well absent in the cell if there's no lactose present. Now, if there is lactose present, a derivative of lactose binds to the repressor. And now the repressor changes its shape, changes conformation, and it falls off the operator. It can't bind to the operator anymore. It's as though this metabolite goosed it. You know, when someone tickles you, you can't hold on to things. If this thing comes up and it tickles the protein, it can't bind to the operator. Now, RNA polymerase is able to initiate transcription from that promoter, and you get these protein products. And now you're able to metabolize lactose. And this will be sustained until lactose levels get too low. Then the repressor will rebind to the operator, and those genes will be repressed again. This is the lactose operon hypothesis. Extensive work on the lactose operon hypothesis shows that this model is largely correct. Largely correct. All right. Any part of that you want me to repeat? This all works because of DNA binding proteins. In fact, Jacques Monod, did his early work on enzymes that change shape when they're bound by regulatory molecules. That's why when he saw this, he said, this is just like these enzymes I used to work on, except this protein's not an enzyme. Turns out that the lactose repressor is a very difficult protein to work with. But when it was eventually isolated, and people were able to get things to bind to it to get it to change shape. It changed shape, and in that changed shape, it could not bind DNA at all. So that's an example of an inducible operon. The operon is repressed, and then when the metabolite, the substrate is there, it's de-repressed. Now, we are going to see that several pathogenic bacteria are actually able to take certain protein products and they're able to turn them on when they get inside our bodies. So Streptococcus pneumoniae, when it's not in our body, doesn't make the genes for making a capsule. Doesn't need to. The second you inject it into a warm-bodied critter, it turns on those capsule-making genes, and it makes a capsule. And that capsule protects it from white blood cells. Same thing with Neisseria gonorrhea. It is able to attach to urogenital epithelia by virtue of fimbriae. But it only turns them on when it needs them. So these are examples of things that are turned on in response to the need of the organism, because small molecules that are available at that time cause de-repression of these operons. So the lac operon is it always on? Is it on when glucose levels are high, lactose levels are low? Is it on when glucose levels are low and sucrose levels are high? Sucrose, yeah, right, I don't think so. Is it on when glucose levels are low and lactose levels are high? Is it always off? Pick your poison. Which one do you think is right? Who thinks A is right? Who thinks B is right? Who thinks C is right? Who thinks D is right? And who thinks E is right? D is the correct answer. It's on when lactose is present and glucose is low. And glucose is low. This is just one example. This is a model system. There are other operons that are regulated in a similar fashion. That's very clear. There are other operons, however, they're not inducible. Instead, they get turned on when the metabolite in question is present, because the protein that binds the promoter and turns it on is not a repressor, but an inducer. So the arabinose operon, for example. The arabinose operon is turned on because RSC, the protein, is not a repressor, it's an inducer. In that case, the promoter is so weak, you don't get any transcription, repressor or not. So in this case, you don't get any meaningful transcription unless the RSC protein binds to arabinose, binds to the promoter, and turns the weak promoter into a strong promoter. So there are several ways to skin the gene expression cat, shall we say. Now, bacteria are not capable of sexual reproduction, but bacteria can still exchange DNA with each other. In fact, bacteria can exchange DNA with each other even between unrelated organisms. We call this recombination. And there is a phenomenon called horizontal gene transfer. Horizontal gene transfer. Horizontal gene transfer means that the organism acquires new genes that didn't come from other bacteria, they came from eukaryotes, or they came from bacterial species that are very different from themselves. Bacteria exchanging genes with each other, that's called lateral gene transfer. Lateral gene transfer. Or it's also sometimes called horizontal gene transfer. They're the same thing. So in this case, they can exchange plasmids, they can exchange pieces of their own chromosome. There's lots of examples of this between bacteria. There's three main ways that bacteria will exchange genes with each other. So the first one we're going to look at is conjugation. Now with conjugation, there is actually a plasmid. This plasmid has an operon called the transfer operon. Transfer operon. The transfer operon encodes proteins that allow for the transfer of plasmids between two microbes. And the earliest plasmid that had this was called the F plasmid. So organisms that had the F plasmid were called F plus. Ones that didn't have it were called F minus. And F plus cells could transfer the F plasmid into F minus cells, and then they both become F plus. The initial contact is made by means of a pilus. The pilus is encoded, genes for the pilus is encoded by the transfer operon. When the pilus makes contact with the bacterial cell, it pulls the two bacterial cells together. Those cells touch, and they transfer the plasmid. All that is mediated by genes encoded by the transfer operon. In gram-positive cells, you have conjugation, doesn't seem to be pilus dependent. You still get contact between cells and you get transfer of plasmids. Now, this statement here is really important. Conjugation is a conservative process. The cell that transfers the plasmid still retains the plasmid. It replicates the plasma while it transfers it. So that means if you have a plasmid that confers a certain property onto a certain cell, all the cells in that population will eventually have that plasmid and they'll have that trait. And I know what you're thinking, I'll just answer it now. Yes, antibiotic resistance can be transferred in this way. Those types of plasmids that transfer antibiotic resistance tend to be called R, capital R plasmids. And if you see R followed by a number of some sort, that's a plasmid that encodes resistance to certain antibiotics. Sometimes these plasmids that can be transferred can acquire pieces of the bacterial chromosome, in which case they're called HFR plasmids, high frequency recombination strains. And now you will transfer more than just the plasmid, you'll transfer also bits of chromosome from another bacteria. This is another way that bacteria can acquire genes. And your book essentially gives you pictures on how this can happen. And our plasmids will bear genes for resistance to antibiotics, and they're very important in healthcare settings. So that's conjugation. Conjugation tends to be the transfer of plasmids, tends to be the transfer of plasmids between closely related cells. and it's a conservative process. Transformation involves the uptake of naked pieces of DNA. The uptake of naked pieces of DNA. Transformation allows the transfer of genes between bacterial species that are completely unrelated. Now, you can do this in the laboratory. You can force E. coli to take up foreign DNA. You make cells that are competent. You do this by putting them on ice and incubating them with calcium phosphide. And then you give them the DNA, then you heat shock them. And they take up the plasma. Very easy to do. I can take it to the lab and show you how to do it right now. You know how to do it. Very easy. But there are several cells that have operons that actually get expressed when foreign DNA is around. They actually take up DNA as part of their way of doing business. Now, part of the reason they take up DNA is a very energy-rich molecule. The bacterium looks at that DNA and it goes, yum. But it can also take up DNA and incorporate it into its own genome. And if those genes provide it with a selective advantage, it will keep them. And there are lots of examples of cells that do this. And this gives them genetic variation, which helps them cause disease better. And Neisseria and members of the genus Haemophilus are two of the biggest offenders in this regard. Very good. We will pick up next week because we don't have class Friday. We'll look at transduction, and hopefully we will finish this chapter and move on to chapter 3. Okay, Tia, get better, get well, drink orange juice, and come back to us well, okay? You have a good day.